If anyone ever tells you that Dr. James Tour is a relevant expert on the origins of life, just show them the rest of this video because we're about to go through and expose the depths of this man's ignorance, deceit, and just overall laziness in his approach to this entire subject on his YouTube channel. For those of you that might not know, Dr. James Tour is a synthetic organic chemist who has a YouTube channel where he does Christian apologetics under the disguise of science communication about the origins of life.
I will share with you about why I embrace the resurrection of Jesus.
Now, why might amyloids be relevant for origin of life?
My name is Grayson. That unedited clip that you just saw was from Dr. James Tour's YouTube channel. And if you're curious about what Jesus Christ has to do with amyloids, biochemistry, and the origin of life, well, you're not alone. Again, I did not edit that clip. It was a hard cut from James saying, email him and talk to him about Jesus so he can try to convince you that Jesus is God. And then it just hard cuts to this other organic chemist that James brought on to do a video specifically about me and the amyloid world hypothesis. It just cuts to that guy just delving right into amyloids. I mean, I could not make this up. I mean, that cut, it's just bold-faced, right? The science, the origin of life stuff is just window dressing for the evangelizing. Dr. Tour has become perhaps the biggest Christian apologist in the world, specializing in abiogenesis, or the origin of life. And just to be clear, Dr. James Tour is a very prominent synthetic organic chemist with tons of publications, none of them on the subject of the origin of life. He's never published or peer-reviewed anything about this area of science that he dedicated his entire YouTube channel to trying to debunk to win souls over for Christ. In fact, as this video shows, he doesn't even read the origin of life research or stay up to date on it. And I think that at this point, I have the receipts to back up that statement. You see, back in May of 2023, I actually visited Rice University to attend the now infamous debate between Professor Dave and James Tour, which ultimately devolved into a shouting match, but was still very entertaining. And I got to speak with Dr. Tour after the debate, where I asked him about amyloids and the amyloid world hypothesis. And I want to know, do you have thoughts about this scientific hypothesis? Have you come across it before? Since, again, he positions himself as an expert in the origin of life, and this is an increasingly prominent hypothesis in that field for the last decade or so. When I asked it to him, he said, no, I actually don't know much about that. So he couldn't make any comment about amyloids. He didn't really seem to recognize much about amyloid chemistry specifically, and definitely had never heard of the amyloid world hypothesis. In May of 2023, I sent him numerous studies of amyloid literature. As well, as you all probably know, I've made multiple videos debunking James Tour's arguments about the origins of life. The last one I made, I thought that was going to be the last one that I did. I addressed right at the heart of why his arguments are fallacious, summarized it all up in that video, and I thought I would wash my hands with it, and I'm done. Because James Tour had never responded to any of my previous videos. That is, however, until 2 weeks ago, when finally, after almost 3 years, Dr. Tour made a video responding to the amyloid literature and the amyloid world hypothesis, and even directly responding to my video on the subject personally. In fact, it was almost 3 hours worth of content of response in total. And instead of going through the amyloid papers himself, James does something that I'm gonna be frank with you, I am not a fan of at all. He brings on Dr. Royal Truman, who he passes off as an expert.
We've got an expert here that can talk about it. And so he's got a PhD, and he can address this. And let me first introduce to you Dr. Royal Truman.
He actually has some publications in creationist journals about amyloid world. So I guess that's a little bit better than James, at least. Although, again, neither of these two men are experts in this field. Neither of them have published in reputable journals about origins of life research. Neither of them have conducted origins of life research themselves. James even going so far as to say, I've brought on an expert with me, Dr. Truman, who is not an expert. Again, just to be very clear. I mean, he has a PhD in organic chemistry, which, again, makes him more than a layperson. He's certainly qualified to talk about the organic chemistry, but we'll see that he makes mistakes approaching a field, an interdisciplinary field like abiogenesis, approaching it as an organic chemist. We're going to see causes some issues that someone from outside the field might be able to have caught if they were truly an expert like they're passing themselves off to be. And part of the reason why I'm making such a point of this is because one of the attacks that Dr. Tour and Dr. Truman level against me in the video is trying to say that I'm a YouTube scientist. Which, guys, I don't know how many of my videos you've watched, but you'll not find a single one where I've ever claimed to be a scientist. Dr. Tour is literally passing himself off as a YouTube scientist. I mean, he doesn't do any origins of life science, but his entire YouTube channel is all about him positioning himself as an expert in origins science. Like, just think about the video that we're going to be watching, right? I mean, James Tour obviously did not know much about amyloids, so he brought on a creationist who had published in a creationist journal that he came across, who said, oh, wow, this guy wrote a whole article debunking the amyloid world hypothesis. That's great. I'll have him on my podcast, so that way I don't have to do any of the reading myself. So it just is like, on top of just being dishonest and passing yourselves off as experts in an area of study where you don't have expertise in and you're not doing active research in, on top of the dishonesty, it's just lazy. To like, James had almost 3 years to actually research the amyloid literature himself so that he could debunk it for like his YouTube channel where he's famous for debunking abiogenesis. Like, that's his whole shtick. And instead of actually taking the time and effort to learn the science himself, he just outsources it to Dr. Truman, who is also not an expert. And his ignorance is very, very apparent. So we're going to start off by watching these two luminaries break down one example amyloid paper just so that we can get an idea of like what kinds of mistakes and errors these guys are making and what kind of game that they're playing that's, again, very dishonest and lazy. And so we'll just go through one paper just to give an example of like what these guys are doing, because they go through a lot of these papers. And then we'll get down to the part where they actually play like a clip from one of my videos and react to it, and I'll just give some of my thoughts. Anyways, because Dr. Tour made 3 hours worth of content about this, I'm going to have to split this video up into parts as well. So I just wanted to get this one out first so you guys get a rough breakdown of some of my ideas. And then in part 2, I'm going to be bringing on some actual experts, hopefully, and a few surprise guest cameos that I think that you guys are going to love. So I just want to get this one out ASAP. The entire setup is that these two phony experts are going to be making playtime peer review on YouTube, and they're going to be trying to tear apart these actually published abiogenesis studies. The very first study that these two are going to be taking a look at and debunking is by Rout, Riek, and Greenwald from 2018, and I actually had 2 of these amyloid scientists on my channel earlier this year to talk about amyloid world and James Tour's criticism, so check that out. James told me that he never watched it. And again, Dr. Tour has never read this study that he's about to be debunking. He's just going to be listening to whatever Dr. Truman tells him about it. He's never read this himself. Let's just listen to Dr. Truman's take about what's wrong with this study, which shows the templating abilities and properties of amyloids.
They took 2 of these peptides of exactly the right length, synthesis in the laboratory, mixed them together, agitated them at 800 RPM, used the right pH, the right temperature, and then waited until they formed the amyloids. Solidification to break them up, make them look nice and pretty. So basically, you've done a lot of lab work here to create the amyloids you want. And then what happens? So you had to use only L-only peptide substrates and templates. Otherwise, you don't get the beta sheets at all.
I see this complaint leveraged quite a lot against abiogenesis research, right? If the scientists' research question, which in this 2018 study, they were specifically interested in, do amyloids have the ability to act as a template for catalyzing their own self-replications in a stereoselective manner? That was the research question. They designed their study in order to answer that research question because that's the way that science works and is done. And I would expect that both of these PhD organic chemists should know that. So the actual study was designed to test this specific property of amyloids. It wasn't designed to test, right, whether or not you can just make these self-templating amyloids arise from scratch in prebiotic settings. Like, there's different studies that look at the prebiotic formation of amyloids under prebiotic conditions. If Dr. Truman or Dr. Tour were trying to make the claim that amyloids are not going to form prebiotically, that's the study that they should be criticizing. The study that was trying to look at whether or not amyloids can form prebiotically. You'd think that they would look at that study. It's by Greenwald et al. From 2016. I know that Dr. Tour should at least know about it because I emailed him that study as well as told him about it in person way back during that debate with Dave Farina. So he should have looked at Greenwald et al. From 2016, which was looking at the actual prebiotic formation of amyloids. And guess what? They did not use these protecting groups. They used prebiotically relevant volcanic gases as catalysts. So then you might say, well, hey, why didn't they use those prebiotic catalysts for this experiment, right? Because this experiment was concerned about analyzing what properties the amyloids have once they're already formed. They were not curious about the formation about amyloids in this experiment. That wasn't the point of this 2018 experiment. And so they start with their amyloids and then they want to analyze what properties they have and whether or not they can act as templates. That was the result that they wanted to investigate. And this leads into probably like one of my biggest complaints about Dr. Truman and Dr. Tour's entire video here on amyloids, right? They are highly, highly selective about which studies they look at. Now, I know that Dr. Tour hasn't read any of these studies, like not even a single one. But Dr. Truman supposedly has gone through this amyloid research to some extent at least. He should know about the 2016 paper about whether or not amyloids can form prebiotically. And that's the paper that they should be going over right now, because in that paper, they didn't use the protecting groups. They didn't use any of the things that they're complaining about now. It's like, okay, they used, I think they only did use a homochiral one for that 2016 paper. And then, okay, you could say, well, then they're going to make that same criticism for that as they did in the 2018 paper. But here's the thing, though. If you're going to be criticizing an entire body of literature, like they are trying to make a criticism video for all of the amyloid world hypothesis, all the literature, not just individual papers. So they need to then check, hey, is there a paper where they've added feedstocks of both L and D amino acids to these amyloid templates and seen, like, if you have anything viable happening there? Because Dr. Truman made the claim just now that amyloids will not form if you have this racemic mixture, right? Oh, if only that there was a subsequent study that was showing the exact data of what happens when you have L and D over time and whether or not the amyloids themselves can get increasing amounts of L over time in the actual empirical data. If only we had those studies. If only Dr. Truman had mentioned, hey, actually there are studies that are showing exactly what I just claimed is impossible. And no, these two creationist scientists make claims. Amyloids cannot do X. Then they look at a study where amyloids do not do X, and they ignore all the studies where amyloids are clearly doing X. That's why it's dishonest. You're supposed to be criticizing the entire body of literature in which there are studies that do exactly what you're criticizing the study for not doing, and you're ignoring them. You're pretending they don't exist. That is dishonest because you're giving people this idea that, hey, the entire field has this problem of using these protecting groups, using this homochiral feedstocks, and it's not true. Like, we have studies where they've investigated this and shown that it's not a problem for amyloids. And these two clowns are ignoring those and telling their audience that these are real issues.
You've got to have very, very precise sequences, okay? Otherwise, you don't get these beta sheets. And they've got to be identical, okay? So you need a very high concentration of identical, very, very rare peptides in, of course, a closed container to keep out contaminants.
Okay, so this part is actually a little incredible because almost every single thing that Dr. Truman just said is factually just wrong. Like, he said you need very precise sequences to get amyloids. Not true at all. Amyloidogenic propensity is very robust. You can have a ton of different sequences that all form amyloids. Like, you can actually just look at any random sequence and then gauge its overall likelihood of forming amyloid beta pleated sheets. And like, yeah, some sequences are more likely to form amyloids than others. That does not mean that you need a highly precise sequence that is literally just lying. And like, he should really know better. I mean, like, straight out of the textbook will tell you that the vast majority of all proteins have some kind of propensity to form amyloid beta pleated sheet structures. Like, straight up, he could not be any more incorrect than the statement that he just said. Until we get to his very next statement, where he says that you need identical amino acids in order to have this amyloid formation. No, that's not true at all. Like, even within their study, right, in the 2016 Greenwald study, where they looked at the prebiotic formation of amyloids, which these two bozos conveniently never brought up at all. But if we look at that study, we can see, hey, they showed clearly in that study that you can form amyloids prebiotically using non-identical amino acids. This has been shown empirically. And yet here we go, the so-called expert is literally claiming the exact opposite of what has already been demonstrated in experiments almost 10 years ago at this point. And then finally he said that this has to be done in a closed container to prevent contamination. Like, dude, like, sure, it's nice not to have contamination when you're doing a laboratory experiment and you need to precisely characterize your results. That's why they don't want contamination. You have literal observations of natural selection coming into play and preserving the uncontaminated form of self-replicators in prebiotic systems. Like, we have the demonstration of that. It works. Okay, it's fine to have contamination because there are error-correcting mechanisms from natural selection in these self-replicating molecular systems. Here's the source for it. Maybe we should just pause for a second and assess where we're at, right? James Tour, a supposed expert and not at all YouTube scientist, an expert on origin of life, had to bring on another expert from outside of this field to talk about amyloids. This expert, in a 20-second clip, just got 3 things, like fundamental things about amyloid chemistry and origin of life research, just completely wrong. I mean, and this is our expert?
I want to let you go on. It's just, this is such a cheat. And to somehow suggest that this is relevant to origin of life when you're doing this selective blocking and activating, it's I'll just call it a cheat. We'll talk about it more at the end, but go ahead.
Yes, it's a good observation. That is correct.
Yeah.
Let's just be clear here one more time in case you missed it. James Tour has never read this study before. He's just letting Dr. Truman tell him incorrect facts about it and then literally just jumping to the conclusions that it's cheating, it's a scam, it's not relevant to origin of life at all, because he's literally just ignorant about the state of the literature. He doesn't seem to know about the 2016 study where they formed amyloids from prebiotically relevant catalysts, like without protecting groups. But then all of a sudden he's just going to claim, well, because this certain 2018 amyloid study that wasn't even concerned or investigating amyloid formation at all, because this study used protecting groups, well, therefore, it's not relevant to the origin of life. Like, if you want to know, Dr. Tour, the real reason why the scientists in this study used protecting groups and why they didn't use volcanic gases for their catalysts, there's some really good reasons for that. And you know what it is? It's really dangerous. Like, I asked Dr. Greenwald about like using carbonyl sulfide, the volcanic gas, to catalyze these reactions, and he was like, "Oh yeah, well, we stopped using that because it's like a huge fire hazard in the lab. Like, we just wanted to show like it could be used to form amyloids, but after that, like for subsequent experiments, we do not want this in our lab for like very clear safety purposes. It just is a huge headache and storage is pain, and it's just like there's logistical reasons why they're not using volcanic gases in their lab, because they've already shown that they can effectively catalyze amyloids prebiotically. Like, they don't need to show it again and again and again. They just showed it the one time in the 2016 paper, so maybe y'all should have started with that one, huh?
They also mentioned that the activating agent that they're using, CDI, shares a common intermediate with prebiotically relevant activating agents like carbonyl sulfide.
And the second reason, right, why they use these protection groups to make sure that the exact, regioselectivity was all checking out, is because of the time, right? I mean, let's all just take a break and think about this. If we're trying to follow the creationist advice, and they were trying to do all of their origin of life research using only prebiotic conditions and only the way that nature actually would have done it 4 billion years ago, it would literally take millions of years. We should all be able to agree. You, me, creationist, Dr. James Tour, everyone agrees. The idea that the scientists have is that this abiogenesis process took millions of years. So if you're going to try to criticize abiogenesis research as saying, well, you're not doing it in exactly the way that it would have happened on Earth in your lab. Because if the scientists try to do it exactly the way that they think that it happened on Earth, it would literally take millions of years. I mean, just like use your head for 2 seconds to think about this, James. Or here's a novel idea. Maybe just read the study yourself, Dr. Tour. Read it yourself before you make a video criticizing it, because it is very, very apparent that you don't know any of the details of the study that you're supposedly supposed to be criticizing in this video. The research question that these scientists were trying to investigate was whether or not amyloids have the property for this self-templating behavior. That's what they were concerned about. That's what they were investigating. That specific little topic. They weren't trying to recreate the entire process of abiogenesis in their lab with all these prebiotic ingredients over millions of years. This is a foolish thing to think, especially from a scientist who has published as many studies as you have, and who should know about experimental design. You should know what their research question was and how they designed their experiment around answering and investigating that research question. If you understood that, then your criticisms here, James, don't make any sense. But again, if anybody was doubting me that James has not read this study, this next clip is probably one of the most damning ones of the entire video, to just expose the depths of just how little this man understands what he's talking about.
So the ratio is that
Yeah.
is that 2.8 to 50 or something?
Exactly.
All right. So you have 2.8 has gone up to 50. That ratio is 50. So in other words, it's. Do they have any idea of the yield here? What does 50 mean?
Trying to remember exactly.
So if you ever hear anyone again telling you that James Tour or this Dr. Truman guy know anything about what they're talking about, just show them that clip. Because this depths of both of their misunderstanding here is really quite remarkable. Like, the entire confusion that they're so confused about, this 2.7 and this 50 number, which, by the way, are not even the actual numbers from the study. It's 2.7 and 51%. This is not a ratio of 51 to 2.7 like James Tour is asking about. I don't know why Dr. Truman doesn't know what these numbers are, considering he made this slide. And on the very slide itself, it labels what these numbers are. They are percent yields for two different reactions. One reaction had a 51% yield, and the other reaction had a 2.7% yield. This is not a before and after. It's not a ratio. Like, both of them just have no idea what they're talking about. The 51% yield was for the condensation reaction of adding in a specific new amino acid onto the template of amyloids in the aggregated form. The 2.7% yield was for the exact same reaction, just happening in a soluble form. So if it's dissolved in water versus if it's clumped up as a little aggregate. Those are what those two numbers are describing. Absolutely nothing at all similar to what either of these two PhD scientists who are passing themselves off as experts, who are trying to peer review this study and are claiming that it's idiotic, it doesn't make any sense, and they literally don't even know what the results of the study are. Like, James Tour is asking, hey, I'm really confused. What's this 50 to 2.7 ratio? Just tell me what the percent yield is. Like, are you serious, Dr. Tour? It's literally, that is the percent yield. Like, how is he missing this? How does he misunderstand this badly? And this is the guy that all the creationists are trying to prop up as an expert? Like, oh, of course his organic chemistry PhD gives him the kind of technical bona fides, the skill set to be able to critically analyze and peer review these origin of life studies. This guy can't even figure out what the percent yield is when it's like just directly reported by the study. I mean, maybe if he was actually reading these studies himself and taking the time to go through this, maybe this is not a mistake that James Tour would make, but like then like the criticism is just that he's way too lazy to be doing this criticism.
Do they have any idea of the yield here? What does 50 mean?
Trying to remember exactly.
The reason I say this is that
Right.
Years ago we had a project where we were trying to make a mother-daughter duplex, something very similar to this, but not using biological entities. And the fidelity killed us. We would have 30% of the non-correct one. And then that non-correct one became a template as a new mother for making other things. And within a couple of cycles, the whole thing is absolutely unusable. These guys are using biological entities. So you'd think that, they're taking advantage of biology, what biology has worked out here. And unless they're going to tell me what the yield is and what the selectivity is, they're just showing the ratio. It was 2.8 before, it became 50 now. So It was higher. But if it's not at 99.9% of the one they want, fidelity's going to kill them. But if they're at 50% of what they want, this is a bunch of nonsense, utter nonsense. So tell me where they are on this thing.
Right. I'm trying to remember exactly.
At this point, is there anything Dr. Truman can tell us about this study? But in all seriousness, this is one of those instances that I alluded to earlier, where I was saying that these two are letting their sort of organic chemistry silos misinform them about the actual study that they're supposed to be criticizing. You see, maybe in James's previous synthetic chemistry experiment, that their fidelity might have killed them, as he said, and they might need these extremely high fidelities for their replications in those settings. all well and good. That's synthetic organic chemistry. But in biology, and specifically amyloid prebiotic chemistry, that is not the case. We already mentioned earlier how amyloids have a robustness to their sequences, and we do have studies showing that they can tolerate quite a high number of mutations, right? Errors in this sequence replication, and still retain function. Remember, this is actually one of the key necessary properties for the origin of life. We have to have Darwinian evolution kick in on a replicating molecular system. And the criteria needed for that to happen include having a population with a large amount of variation in their fitness effects. So, when you have a chemical species like amyloids, which are capable of these self-catalyzing replications, that can tolerate a high mutational load and that generates a large amount of diversity in these different sequences, what happens? Well, some of them are going to be more stable than others. Some of them are going to be faster at replicating themselves than others. You have this generating mechanism for variation that is actually required, one of the necessary criteria for the origin of life, because it's needed for Darwinian evolution. Another thing that Dr. Tour seems to be misunderstanding is that he seems to think that these are under equilibrium conditions. Maybe because typically in his lab, maybe that they would be under equilibrium in his experiments. I'm not sure, but in this particular chemistry of amyloids and the origin of life, this is far from equilibrium conditions, and I don't think that James necessarily realizes that. The products, the amyloids, are some of the most stable forms of proteins that exist. They can literally last for years and years and years out there in nature today. So they're not going to be back reacting and going back into the reactants like at equilibrium. So James just seems to be misunderstanding just where we're even at with the biochemistry here.
In the case of phenylene, it's 85%, so it is real.
So
It is real.
But you have 15% going in the wrong direction.
Yeah.
It's dead. It's dead. I mean, if
I know.
15% of these are going the wrong way, it's over.
So this is just an instance of James's previous experience as a synthetic chemist leading him astray, and his lack of experience in biology in general and in amyloid chemistry in specific, causing him to make some very, very incorrect claims here about needing 99.999 whatever percent fidelity, which, like, any biochemist, any amyloid chemist, would be able to tell you is just kind of absurd. We have studies looking at these self-replicating chemical systems to see if there are these sort of natural selection error-correcting mechanisms that come into place, and yes, they are empirically observed. Here, you can just read the study yourself, but I doubt that he will. Does anyone else find it to be just so ironic when Dr. Truman specifically calls out YouTubers, like insinuating, talking about me, as saying that they don't typically read the studies that they talk about, in the middle of a video of YouTuber James Tour claiming to be a scientist peer reviewing a study he hasn't read? Come on, how is the irony just going right over Dr. Tour's head when he says that?
Unfortunately, 99.9% of all YouTubers don't read technical papers.
And Dr. Truman even goes so far as to correct James on this whole 99.99% yield claim of his. He's like, well, Dr. Tour, I don't really think that that's the main problem here. And then he very carefully and politely tries to massage the conversation in a new direction.
So it is indeed a high yield, but that is not really the fundamental problem. You're right that there are some serious problems with it, several problems, but and in fact, you're alluding to it correctly. It's a yield problem, but the problem is actually a little bit different than you suspect.
So let's see what Dr. Truman's whole big issue with the study is, and then let's see whether or not James even agrees that it's a problem.
This plus this plus the amino acid are all together at the same location. That means that
yes, they've
right?
conveniently put everything in the same reaction tube
Right.
to react, and the
Exactly.
things are not like this. But we'll give them that, even that. But now you have these conveniently activated. This is huge, huge human input that would not be available on Origin of Life. This is a bunch of nonsense when it's applied to Origin of Life. And I'd love to see the yields here to talk about the fidelity of this, because if it is not very high purity, this whole idea of templating and reproduction is toast. It's over. But anyway, press on.
Okay, so Dr. Truman's big problem seems to be that all of the reactants are all together in the same reactant vessel, and like maybe like he was trying to get to some sort of issue with the concentrations or something. We can talk about that later, but James just like immediately cuts him off to say, but my yields, like he just cannot drop the whole like percent yield thing, and it has to be 99.9. Like he just has to go and make that point again and derail Dr. Truman. Like they're like just literally butting heads here. Like they can't even agree about like the problem with the paper that they're supposed to be debunking that James Tour hasn't even read.
Over. But anyway,
I'm with you, but let me be fair to them, to at least make sure that we all understand what is being argued, and then afterwards go into the plausibility aspect, because there's several issues here. But it's important that we first understand what they're trying to do.
Somebody needs to give Dr. Truman an award. Like, the cordiality, the manners, the politeness that he just expressed is truly impressive. I mean, he literally he got derailed by James, who wants to bring up the stupid yield thing that Dr. Truman already tried to like have him save face on and try to move away from to a different topic. And James goes back to the stupid claim about the yields. And Dr. Truman is just like, yeah, I mean, I think maybe we should focus on like what the scientists were like trying to do here with this experiment. I mean, Dr. Truman is being so nice here, and James is just bulldozing his way right through it.
This would be laughed at. I mean, it's laughable because you're doing this multiple times, so you keep inserting 30%, 33% of the wrong isomer. Every time you've inserted a wrong isomer, boom, it's done. It's over. So this is not a templating effect. I mean, it's so poorly done that this is not a daughter from that mother. This is another molecule. So this whole idea that you're copying, you're not copying at all. If these guys don't have the pure amino acid, the enantiopure amino acid to start with,
right,
it's toast again. So this is another reason why this is nonsense. Go ahead, press on. I'm just trying to just give a little short critique here, but I don't want to slow you down too much. Press on.
That's true.
Because of you, millions of people have heard the gospel.
Again, I just had to include that in there unedited from the original footage because like this man is literally like a cartoon character of himself at this point. Like, this is the third time that he has bulldozed over Dr. Truman to bring up the same point about the yields again. Only this time he's talking about the L versus the D different isomers being added. And if you add the wrong one, for some reason, Dr. Tour thinks then it's just a dead end. Then it's nonsense. Then in the whole study, we should just throw it out. And it's a laughing stock. It's like, I just desperately wish that someone would show Dr. Tour the study that I already showed earlier, where they just did this exact thing in the lab, giving both L and D amino acid building blocks to the growing amyloid, and they saw over time, like, you're seeing it right on the graph from the data that they measured themselves, there is a very strong preference over time for the amyloid to selectively add just one type of isomer and not the other. Like, we can see this happen. Like, over time, the percentage of D versus L is more and more and more different. Like, the actual data, the empirical reality, just directly refutes the nonsense claims that James Tour is just making for over and over and over again at this point.
Here's the problem. These substrates are L only, right? So here's a 7 amino acid L only. And when you've got all of these L only, it forms a structure that actually decreases the ability of another L from adding to that particular substrate. It decreases it.
When an amyloid is acting as a template, there should be a much stronger preference for being enantioselective for just an L or just a D, depending on the template, versus if it's just an amyloid that's not acting as a template. This is all like explained in the paper. The data's pretty clear. It's like Dr. Truman telling his own misunderstanding to Dr. Tour as if it's like a fact that he's uncovered in the data that the scientists are hiding.
We're getting less L than you want by chance being added to these substrates. And is that significant? And that is enormously significant, even though it wasn't mentioned in the paper, but the data is showing this.
Actually, Dr. Truman, it's explicitly mentioned in the paper where they specifically highlight where the soluble amyloid favors the D enantiomers for these certain amino acids, such that when it's templated, the actual template amyloid inverts and enhances the stereoselectivity for the L enantiomer. So this is just showing how good amyloids are at templating these stereoselective reactions. This is why, if you're gonna criticize a scientific paper and try to debunk it, Dr. Tour, you should always read the paper yourself first, because otherwise, Dr. Tour is just gonna take Truman's misunderstanding and run with it.
You end up decreasing the chance of adding another L to it.
Okay, so let me just cut to the quick here and just give the explanation of this. This is tremendous. How anybody can look at this and think that this is relevant to origin of life is just, to me, absolutely idiotic. Here, what he's showing then is that if you have the all L, which is what you're going to need for the natural system, it disfavors being a template
Right.
for another all L configuration.
Exactly.
It actually is favoring the opposite antipode, the opposite
Correct.
enantiomer from coming in. That's what's favored. So again, this argues against this being relevant to the origin of life. This very data, their very data argues against it. This is what I say over and over again. I look at the same data and I say, that tells me that's not how it happened. This is not how it happened. So anybody who would look at this and study this and look at this, as you have said, you'd have to say this argues against the origin of life. Anyway, I digress. No, I don't
No,
digress.
that's exactly right.
No, that's incorrect. What the data is actually showing is that the addition of stereoselective amino acids is not likely to proceed in a soluble substrate form when it's not in an aggregated template. So the actual data is showing that when the amyloids are not solubilized, when they're aggregated together and they can start to template each other's additions, those templated additions are extremely stereoselective when compared to the control soluble substrate form. That's what that data is showing. And both of these guys are jumping to conclusions that are entirely unfounded by that data. Like, they're seriously trying to make the argument that if the soluble solo amyloids that literally had the 2.7% yield and like barely any enantiomeric like excess, just like a little bit in the D enantiomer favor, because of that, we have to then rule out the where we see the aggregated amyloids coming together and templating each other's reactions in a way that can like reverse that trend entirely and then amplify the selection of just the L amino acid additions. Okay? So because the control is not exhibiting the same templated selective behavior as the aggregate, the actual thing that we're trying to discern the behavior of, then we have to throw out the entire theory that these amyloid aggregates templated their own self-replication to reproduce like homochiral sequences. Like how does that follow?
The point is, this says absolutely nothing about how life could have originated. Quite the opposite. It's just another example of what could not have happened. You know, you're kind of running out of ideas eventually. But I don't want to criticize the cleverness of experiments because, you know, chemists
I'll criticize it. I'll criticize it because this clever experiment is actually teaching people, is actually suggesting to people that this is how it happened when it's talking the opposite way. So the poor little YouTuber,
hey, that's me,
who thinks that this solves the origin of life problem, he's going ahead and putting out all these YouTubes.
By the way, if you like this YouTube, please like the YouTube and check out any of my other YouTubes on my YouTube. Thanks.
And the poor guy is so deceived. That's the problem with it. And then he deceives everybody who listens to his content.
The amount of projection going on here is absolutely insane. Like, Dr. Tour, who just got deceived into believing something untrue about the study in question from his guest, and then went on to deceive his own audience about it, is going on his pulpit right now talking about how I am the one deceiving my audiences. Right.
I mean, this just condemns it. It condemns it, so I'll go after them. I'll go after them.
That's great, James. Go after them. But if you're gonna go after them, you might want to just start by, I don't know, maybe reading the study first. Maybe read what they wrote before you go after them. Doesn't that make more sense? I mean, I know that you've peer-reviewed studies before in organic chemistry, James. How many of those studies did you peer review without even reading first?
Okay, let me just give a quick summary here. They had to have the protection, deprotection, the protection activation. This I described in my 14-part series, where you have to have protection to be able to do this. So none of this will happen on an early Earth. There's no selective activation like this. That's number one. Number two, it was totally loaded based on the specific amino acids that they were using. You have the wrong amino acids there, it's not going to work. It didn't give the stereoselectivity. It didn't pick out the right enantiomers. It didn't give the regioselectivity. They had to use a non-canonical, a non-natural system because the natural system didn't work. So they used an unnatural system, and that unnatural system worked only poorly. When 15% would have cross-linked, you have 1% cross-link, and the whole thing changes. If you have a rubber band and the polymers are not cross-linked, the thing is a liquid. And you put 2% cross-link, you get a rubber band. I mean, cross-links change everything. And in peptides, you get these cross-link, it's game over. They had the concentration. They would have had to have a concentration where they would have had the sky raining pure amino acids to get that kind of concentration in the oceans or in some tidal pool to do this type of thing. They adjusted the pH. The pH has to be just right. You can't have too much acid. You can't have too much base. It has to be just right. I mean, these are Goldilocks conditions. And then the temperature had to be just right. and so they adjust all these things. And the stoichiometry had to be just right, meaning that they couldn't have too much of one versus the other. They got the stoichiometry just right. And it only worked with specific sequences. So you couldn't just have your random amino acids around. This whole thing is loaded. Dr. Truman is very gracious man. He says they're clever. Yes, they're clever, but they're cheating. They are cheaters. These people are cheaters if they are relating this to an origin of life experiment. And the victim of the cheating are these YouTubers who take it and pick up this banner and say, look at this banner. And they run around with this banner as if they've got it. The poor folks are victims of the origin of life community making these claims. That's exactly what I've been saying all along. Okay, sorry for the interruption. I just had to have people understand this.
Yes, poor little old me and my YouTubes. I've just been victimized by these mean old lying origin of life scientists. Oh, I'm such a victim, aren't I? Well, according to James Tour, anyways, I am. But in all seriousness, I already have debunked most of the points that James just brought up there, like from like him misunderstanding the points about, oh, if you have a crosslink and then like one particular amyloid product becomes a dead end, like that's fine. These are populations which have lots of variations in them and are being selected for based on their like replication rates and their molecular stabilities. Like it's fine to have dead ends. These are akin to like mutants that have really low fitness in modern evolutionary populations. Like James just doesn't seem to understand this. I don't know if he just cannot think like a biologist or what it is that is the disconnect here. But the other things, like he's saying, oh, the pH has to be so precise. Like there are proton gradients that are naturally occurring at hydrothermal vents where a lot of this chemistry is thought to take place. So there are little gradients of pH right there. So it's not like it has to be fine tuned. Like the reaction will occur like in the gradient where the pH is suitable. Same thing as a temperature. This is a temperature between like open ocean and like the hot volcanic gases escaping out of hydrothermal vents. Like there are gradients of these things. They don't have to be finely tuned in the primordial earth like they do in a lab where the chemist is trying to only measure one particular aspect of a reaction over a very small time frame, right? We already made the point that if James had his way and all the scientists for like, if they wanted to check to see if amyloids had templating abilities or properties, instead of just look at the amyloids and see whether or not they display those properties like the study actually did, if we follow James' advice, the scientists would have to first start with only prebiotic reactants in like an uncontrolled reaction area, not even a container, because apparently that was an issue too. So it has to be out in the ocean somewhere at a hydrothermal vent, and I don't know how the scientists are supposed to record their results or measure anything because it's so chaotic, but according to James, it needs to take place with only prebiotic conditions, not even in a container. And I mean, like, it's going to take millions of years. So I don't know if he wants to sit around for that, and if James will just say that we're clueless until we just evolve life over millions of years in some sort of enormous cordoned off hydrothermal vent experiment. Like, what are these people actually wanting from origin of life experiments? Like, I hear that they have all these problems, but like, can they ever do some constructive criticism here? Like, what does James Tour actually want to see from any of these experiments? He never seems to say, does he?
When you put your cleverness into these experiments and it still doesn't work, it works poorly, and it could never have shown the fidelity that you needed, never could have shown the coupling that you needed. You set up one condition after another in addition to the concentration, and it still doesn't go the way you want it. That's really bad. And then to suggest that this has something to do with origin of life, liar, liar, pants on fire. That's what they are. Go ahead.
Yep.
Sorry about that, Dr. Truman.
No, I understand.
It makes me think that it was definitely Dr. Tour's idea to bring up my video next to react to in the second half of the video, which, you know, I guess I'm flattered that Dr. Tour felt like he needed to bring on another PhD so-called expert just in order to, like, react to my video. So, I mean, that's kind of cute, I guess.
They choose this 50-second clip at the end of my Amyloid World video. And so let's see, they're going to pick 6 specific things that they say that I got wrong in this little 50-second clip, and then they're going to debunk me. So let's see how that one goes.
The 50-second clip begins with a statement.
ATP is another naturally occurring energy source near hydrothermal vents.
Okay, that's just a statement of fact. That is totally nonsense.
Karma has come to collect. They got me. I have to take this L, all right? What they're saying is correct. On a prebiotic Earth, ATP would not be naturally occurring that we know of at hydrothermal vents. At least there's not direct evidence to support that. I did misspeak, and I will own that. Like, when it's been brought to my attention that I have said something incorrect, I'm happy to admit that. Like, I think this issue was mostly from the fact that these videos are just me extemporaneously speaking, which, I could definitely see the issues with. Like, if I were to actually take the time and script out everything I was going to say here, that mistake would not have occurred. But I would also like to point out that, right, they're supposed to be debunking my video about the amyloid world hypothesis, and, like, they've skipped past the entire video that is about the amyloid world hypothesis, and they skipped to the very end where, in the format of the video, at the end of the video, I wanted to speculate about how we get from amyloids to how we go to, getting towards protocells and life. So I just think that it's really, like, bizarre for them to be criticizing, like, something that is really has nothing to do with the amyloid world hypothesis. Like, nowhere in the amyloid paper did it say that ATP is naturally occurring. I mean, that was just my mistake because I was just talking unscripted. So actually, I am curious. If you're watching right now and you do think, like, Grayson, is irresponsible of you to be doing science communication just with, speaking off the cuff, and that you really should be scripting your videos. I mean, I'm open to that kind of criticism because, I opened myself up for this line of attack right here because I misspoke because I wasn't scripting it. So, let me know what you think. I'm genuinely open to feedback on this, and it would be, a lot more work to script through the videos, but maybe it would be worth it because, like, everybody misspeaks sometimes. I mean, if James Tour never said anything wrong, okay, well, maybe then we would have a point here. But don't be throwing stones in the glass house because just in this one video, think about all the things that James Tour has said that were just factually incorrect. If we're just gonna nitpick every time somebody misspeaks when they're talking extemporaneously, I just don't think that it's like a really good way to push the conversation forward. Let's substantively critique the actual, like, positions of amyloid world hypothesis for origin of life, right? Okay, let's move on.
And once you develop this interaction between amyloids and ATP, it's only a matter of time until you start having refinements into the information storing system.
Like, what? So you've got an amyloid, you got ATP, and suddenly you got a steadily improving information storage system. It's like, scratch your head for half an hour trying to figure out, like, what?
What on earth is he talking about? Help me out here. Help me out here, Dr. Truman.
I think he read, misunderstood some things.
Okay, I see that my imprecise language really is starting to bite me in the rear end on this one because, again, this was meant to be the part of the video where I make a concluding statement, start speculating about where it goes from there and how we start getting to a protocell. That was my intention in the video anyway, but clearly, I was not expecting my words to be dissected every sentence by sentence. But if I can explain what I was trying to allude to, right? There are studies showing that ATP binding to amyloids stabilizes both of them. And these studies from the similar labs from Riak and Greenwald have shown that, like, these ATPs and these amyloids that stabilize each other preferentially bind in triplets, like the codon pairs. And ATP is a nucleoside triphosphate, which is essentially like the building block material from which DNA is created from in modern biochemistry. You take ATPs and GTPs and you string them all together into A's and G's in DNA. And so my point was that isn't it interesting that we see these nucleoside triphosphates preferentially binding with amyloids in triplet pairs, just like they are found in the genetic code. And then we also see experiments with the dynamics of nucleobase and amyloid hybrids, and we see how they not only replicate the sequence of their amyloids, but they also replicate the sequence of their nucleobases that they're associated with that just kind of get carried along for the ride. And so you actually are getting a replication of the sequence information of both the proteins and the nucleic acids, which eventually becomes, the genetic material. That's what our genetic material is today, is the sequential arrangement of nucleic acids. I was trying to speculate at the end of the video based on some new and cutting edge science, which, like, the whole ATP and amyloid interaction thing is still cutting edge as we speak right now. So, like, none of this stuff has been worked out. It was not my goal to tell people that this is how it happened factually. I don't think that's how I was conveying it if you don't just clip it out in like little bite-sized clips here, but okay. They also were trying to equate amyloid's ability to hydrolyze ATP with like literally just dumping gasoline on a car and lighting on a fire and then saying that it's doing work.
Let's say you dump fuel on a car and you put the car on fire. It doesn't do useful work. It's heavy energy or in any way catalyzing the degradation of ATP, releasing a little bit of energy. That doesn't create work.
I don't think that that's a very good analogy at all. I was just trying to give an example of one of the catalytic activities that amyloid is known to do. There's multiple other ones that I could have done. There's myriad examples. There's a paper right here going through several of them, a review article. I would just suggest Truman check that out if he thinks that any of the catalytic activities that are being attributed to amyloids are in any way analogous to lighting a car on fire. Again, and I don't think that either of these guys would be nearly as confused as they both appear to be if they were actually staying up to date and reading the literature in abiogenesis. I think that they would probably know what I was talking about if either of them were more read on this subject.
This nonsense that time refines things. No, it doesn't refine things. It doesn't. Time works against you. If you get a memory stick
Right.
has flash memory. That's guaranteed for 10 years. Why is it only guaranteed for 10 years? Because those bits of information start getting lost, and any memory system, whatever it is, begins to get lost over time. It doesn't refine this; it gets worse over time. And I mean, this is contra-thermodynamic, what you're saying here.
Exactly.
Wow, James. This late into the video and you're literally hitting us with a evolution goes against thermodynamics argument. Are you serious? Like, this is the most low-grade creationist argument that even like the major creationist outlets try to beg creationists not to make these stupid arguments. I cannot believe right now that this is what they're trying to argue. Like, this is some sort of like genetic entropy, like things decay over time, like not understanding that the Earth is not a closed system. Like, it's just that simple. Maybe this is the issue with even arguing with creationists on these highly technical subjects, because you're right here in the weeds of the biochemistry, and all of a sudden they hit you with, oh, well, we all know that over time everything decays and that nothing evolves. It's like, dude, like, if you know how natural selection works, then you know that things can evolve over time. Like, things can get more complicated over time. There are even there are non-evolutionary mechanisms for increasing complexity over time, especially when you're at like non-equilibrium or like near equilibrium conditions. Like, this is just pure ignorance from James right here. Like, I literally cannot even believe that he's trying to make the argument that things cannot get more complex over time. Like, he's just denying that evolution and natural selection even exist or could possibly be relevant for the origin of life. These chemical systems would have to evolve. They would have to have a population with variable fitness effects that's being selected upon by the environment. Like, that's how this works, James. It's going to get more complex over time because that's what the evolutionary forces and selection pressures are driving it to do. Like, this is just such straightforward biology 101 type stuff here.
Anyway, press on.
The stringing of ATP and different triphosphate nucleotides together into DNA and RNA.
The stringing of ATP and different triphosphate nucleotides together into DNA and RNA. Well, um, it doesn't quite understand how the chemistry really works. The truth is, in cells, you do have DNA and RNA polymerases. All right, these are these monstrously complex enzymes. They indeed do use nucleotide triphosphates, okay, as substrate. That is correct.
Okay, cool. I was correct. That's exactly what I was talking about. Thank you, Dr. Truman.
But that is not how this would work in nature and water, and it's not the way origin of life chemists try to produce RNA and DNA. He's got his chemistry misunderstood.
Yeah, so what Dr. Truman is talking about are how origin of life researchers create RNA in like RNA world hypotheses where they're trying to prebiotically generate RNA from like various prebiotic ingredients, and they're obviously not going to be making them out of nucleoside triphosphates. That's not what I was saying or implying, because we're not talking about RNA world hypothesis in this video, are we, Dr. Truman? Right? I mean, like, come on. We're talking about amyloid world hypothesis, and we were just talking about how amyloids preferentially bind triplets of ATP into stable conformations where they're right next to each other and can do a lot of interesting chemistry. Like, that's what I'm alluding to, about how amyloids could facilitate the generation of RNA and eventually DNA. I was speculating. Fair. I think that I made it pretty clear that I was speculating in the context of the video, but yes, I was speculating based on the evidence of what we've seen from the interactions of ATP and amyloids and what it seems to suggest about triplet codons and how they might have arisen. Again, I wasn't talking about RNA world or how biogenesis researchers form RNA without amyloids. Like, that's clearly not the context of what I'm talking about. So what we're hearing from Dr. Truman is a lot of hubris and thinking that he understands and I do not, but in actuality, we see that Dr. Truman was the one with the misunderstanding all along, to no one's surprise.
So you do need special enzymes for this to work. In reality, origin of life, chemists, what they do is they activate the monophosphate, okay?
Well, that's not what the amyloid researchers do, Dr. Truman. Clearly, you are ignorant of the actual state of the literature because you're talking about RNA world research. So, like, Truman does not even know what he's talking about here, clearly projecting that onto me. I'm not talking about RNA formation without any help from any proteins. We're talking about amyloid-assisted RNA formation, or at least we're speculating about it based on the current rudimentary state of the evidence showing the interaction between ATP triplets and amyloids. That's what we're talking about, in case Truman seemed pretty lost.
So not to belabor the point, simply his chemistry is wrong. Let's just drop it, okay? It's not right.
The incorporation of lipids, which are again found naturally at hydrothermal vents, and which spontaneously form bilayer membranes, as well
Then
as
the incorporation of lipids, which are again found naturally in hydrothermal vents, and which spontaneously form bilayer membranes. So now he's trying to go to something that looks like a cell. So you've got a bunch of amyloids and then a bunch of lipids floating around, and now you've got bilayer membranes. It's like, what are you talking about? Like, what is going on? What lipids are being incorporated into what? What are you talking about?
What he's suggesting is that you can get these lipids coming out of hydrothermal vents, and you can. Lipids can come out of hydrothermal vents.
Thank you, Dr. Tour. I mean, he had to educate Dr. Truman here about prebiotic lipids, which do actually naturally occur at hydrothermal vents. So thanks, Dr. Tour, for having my back and saying I was correct on that one. What Dr. Tour and Dr. Truman appear to be ignorant about, where they're just saying, oh, these lipids just happen to just come along and just I don't know, just happen to enclose these amyloids. No, like we actually do have the studies showing that how these things happen. Like we have studies characterizing the interaction between prebiotically relevant lipids and amyloids, and guess what, they stabilize each other, and the amyloids form in like the hydrophilic and hydrophobic areas of the lipids, like through the membranes, and like start doing a bunch of really interesting protocell type chemistry. We actually have the empirical data characterizing this. Here's the studies.
If you get a lipid, it would not be a diasteal lipid, so it would be very unstable. Plus, it would be extremely leaky, so you're not having the proton gradient that you would need. So in other words, it is not a protocell. These things that are coming out of a hydrothermal vent do not give you the protocells that you'd like to be able to call a protocell. So he's got that wrong as well.
So again, Dr. Tour is just ignorant of the amyloid literature. Maybe he should read a little bit more in this area, because then he would be able to have seen the studies that show that the amyloids and the lipids actually stabilize each other, and the amyloids form those membrane protein-like structures through the lipids and can do a lot of this really interesting chemistry. So like James is just flat out wrong in what he just said, because he's ignorant of the studies showing exactly what he just said can't be shown.
As well as the creation of protein enzymes to do specific functions.
So somehow you've jumped from amyloids to now you solve a problem original life chemists have been unable to solve, and that is, where do protein enzymes come from?
So he just pulled that out of thin air, right out of his hat.
Exactly.
No, I didn't. We established that amyloids can have a lot of enzymatic activities and catalyze a large myriad of reactions, and they're going to be producing like multiple daughter amyloids that can then take on these specialized roles. Like, yeah, this is not just pulling it out of a hat. It's like further and further specialization and complexification of these replicating amyloids.
Let me break this down as simply as possible. All of the building blocks that go into the formation of amyloids have all been shown to be naturally occurring.
Now, the claim that all the building blocks that go into formation of amyloids have been shown to be naturally occurring. Okay, let's stop with that. Unfortunately, 99.9% of all YouTubers don't read technical papers, but these kinds of statements are easy to remember. Oh, everything is naturally occurring and you get amyloids. That's easy to remember, and they're simply crazy. Look, they started off with homochiral peptides that were capped on both ends, and the amino acids were artificially activated. There's nothing naturally occurring here at all. The peptides used constantly rely on amino acids that evolutionists believe did not exist periodically or in trace amounts. They rely on this.
Yeah, so Dr. Truman is either ignorant or lying here. Like, he might be able to point to like specific examples of like maybe the 2018 study that they're showing used protection groups, right? But then he makes the claim one step further, saying that all the studies need to have protection groups, which is just ignorant of Greenwald et al. 2016, where they didn't use them, right? And they used a prebiotically relevant catalyst. They didn't use any of the things that he was just criticizing. And you can go to their other studies where they've used racemic mixtures of amino acids and not started with homochiral reactants. So again, the critiques that he's leveling against individual studies, which are irrelevant to their actual research question that they're investigating, he's then extrapolating to try to apply them to the entire state of the body of literature for amyloids, which is just a lie. Like, what he's saying is factually incorrect when you're looking at all of the literature. Like, we can look at that 2016 study and see their prebiotic formation of amyloids in exactly the way that he just said has never been shown. And let me also just point out that in that 2016 study, all of the ingredients that they used to get their amyloids have all been demonstrated to be naturally occurring on prebiotic Earth. So, yeah, I was correct in what I said. So, Dr. Truman, sorry, you failed to debunk me on this one because I was right. Amino acids naturally occurring, carbonyl sulfide naturally occurring. What I said was factually an accurate statement.
So you have these amyloids being formed with essentially random sequences.
He says amyloids being formed with essentially random sequences. There's nothing random about the sequences. These very clever chemists engineered to work.
Yeah, allow me to clarify, right? As we've been over, amyloids conform with a huge number of different potential sequences. Like all manner of different sequences of amino acids could form together into amyloids, right? Now, some sequences are going to be more prone to forming amyloids than others. So yes, it's not truly random. But in terms of like just whatever amino acids are present in the environment that come together to form amyloids, that's like essentially random. That's what I'm talking about. Like, yes, some sequences might be more common than others because they're more preferentially formed, but like there's such a huge amount of different potential sequences that can all form amyloids that it's like essentially random which one that you would be getting. Like that was my point, and that point is still correct. And then just to show off how correct my point was, Dr. Truman is going to provide a citation from CPJ Maury's review article on amyloids, essentially saying the same thing.
That's where he got it from.
Yeah,
That comes.
That's probably where he got it from. Yeah,
he got it from Maury.
Uncoded short peptides, and then you get this error-correcting replicative cycles generate amyloids that represent the first coded peptide polymers. You might as well pull that statement right out of the hat as well. I mean, where this researcher got this thing? Is this Murray again?
Right. This is Maury. I see. Look,
boy, this guy's a piece of work.
Okay, look. Let me. We got to be objecting fair about this.
Whoa, jeez. Did anybody else just detect like pure, bitter, angry hatred right there in the way that James just said, this guy's a piece of work? Like, that was aggressive. Like, dang. I mean, look, I understand if James thinks that he has scientific disagreements with Maury's phrasing there, but like, Maury's phrasing passed through peer review and was published in a reputable journal and reviewed by actual relevant experts in the field. So if James Tour has such a big problem with the information found in this peer-reviewed study, I'm going to need a better justification for why it's incorrect for Maury to say this than just, wow, that guy's a real piece of work, huh? I mean, can you provide any rational justification for why what Maury is saying is incorrect, James?
And then the poor YouTuber takes it as gospel.
Exactly.
This is the danger with you guys. You guys are going to you say these things, look at what it catalyzes. This is the problems. Maury is actually, he is the guilty one, I think. He is the guilty one for having even written it. That you can imagine this? How can you imagine this? You can imagine all sorts of things. You can imagine, people just dropping out of the sky. I mean, you can imagine it. I mean, what does this mean?
Right. And then the last one, I mean, I don't want to belabor the point. I mean, it's a very short video, and believe me, there's a lot worse stuff out there.
Of course he doesn't. And with that, there's just the one last paper that these two covered at the end of their video, but it's like, oh, just going through this has been so long already. We're approaching 2 hours now on this video, and I've only on to their second paper that they've been through in their first video here. So let's just get through their central criticism of this paper and wrap this up, shall we?
What you really have to do here is look at the probability of getting the right peptide sequence. So just to give you a quick calculation, when you look at the relative proportion of amino acids that are thought may have been present abiotically, Look at those proportions, and that gives you then the correct probability of getting a single one of those sequences, right? So these are the kinds of probabilities, and now you've got to have these kinds of concentrations, right? All, of course, homochiral.
So, to the surprise of absolutely nobody at this point, almost everything that Dr. Truman just said was factually wrong. As we already went through, you don't actually need all homochiral feedstocks of amino acids. You can actually have amyloids forming of racemic mixtures, and they just aren't going to be very stable until they start to be selected for based on their stability for more and more homochiral amyloids. And like we saw the studies showing this. So he's just talking out of his you know what right there. And his whole calculation here just shows the true depths of this man's misunderstanding, because he's under the impression that we're only looking for one specific target desirable sequence of amino acids. It's like, have you not been keeping track of literally any of the amyloid research from the past like 20 years that have shown us how sequence robust amyloids are? You're not just looking for one particular sequence and then calculating what are the odds of you forming that sequence from only homochiral amino acids. It's like, this is nonsense. This is a meaningless number that he's calculated that is nothing but a pathetic straw man of what the actual science is even proposing, right? Like, he's calculated one potential sequence forming randomly when he needs to add that over and over and over to all of the potential sequences that would form amyloids, which he hasn't even attempted to do at all. And like, that numerator is just going to keep growing bigger and bigger and bigger, because newsflash, there's a large number of potential sequences that will form amyloids. And this is something that Dr. Truman just seems either utterly incapable of addressing, or he's just like ignorant of it even being a fact, which is somehow worse. Like, this is one of the most basic facts of amyloid chemistry as it relates to like prebiotic biogenesis. So like, if he doesn't even know this much about like the sequence robustness of amyloids, like, then he really is clueless. And yeah, I realize now that I'd said that we would talk about the concentration later, but like, this is kind of our last clip talking about this last paper, and I thought that we had a segment where they were specifically talking about issues with the concentrations being too high for the amino acids, so I want to address that just here at the end. Sorry I didn't find a way to naturally bring it up in the video, but I really did think that this was probably, of all of the criticisms that these two threw out there, right, and they threw out a lot of criticisms, this one I think is the strongest, right? The fact that the concentrations of amino acids are like pretty low in the prebiotic oceans, right? Like, we see them in decently high concentrations in meteorites and things, but in terms of what kinds of concentrations they might be found in and around hydrothermal vents in the prebiotic earth, it's probably lower than a lot of these amyloid experiments. And so that is legitimately one of the better criticisms that they should have absolutely focused on. But again, I'm sure that they would have done it in a way that ignores the gestalt of the literature and just hyper focuses on these studies and say, oh, well, we have to throw it out because the concentration's too high. But hold on now, because I wonder, is there any kind of prebiotically relevant natural concentrating mechanism that might well be available in and around hydrothermal vents exactly where we're saying that this chemistry might have taken place? Oh, yeah, right. There was that study that was showing exactly such a prebiotically relevant concentrating mechanism, where you have the interface of the hot gases coming out of the hydrothermal vent and they're making contact with these evaporating water in the ocean. They didn't look at amino acids in this particular study. They were looking at nucleobases, but they were finding 30-fold increases in concentration right at these interfaces, which is where all this interesting chemistry is probably likely to be taking place in the first place. So this is very exciting. And I happen to have some insider deets about how the authors of this study with the concentrating mechanism actually know a lot of these amyloid authors, and they're kind of right now currently working on integrating this into the amyloid studies. And so we are going to see whether or not this naturalistic concentrating prebiotic mechanism for increasing the concentrations of amino acids to catalyze amyloid formation prebiotically. Like, they're currently doing this research right now with this naturalistic prebiotic concentrating mechanism. So yeah, even if like these two had focused more on the issues of concentration, which like would have been like the most good faith criticism that they could have probably levied, there are still some really, really promising avenues of research that we're currently looking into right now as a scientific community, right? And yeah, these guys are totally ignorant of the actual state and status of origin of life research or what is going on right now. They're not reading any of these studies, as is clearly shown. So yeah, I just find it to be hilariously ironic that in this video where James Tour purports to expose the YouTube scientists, that is exactly what he ended up doing. He ended up exposing himself to be a fake YouTube scientist who pretends to be a fraudulent expert in the origin of life research when he never actually reads any of this research, doesn't familiarize himself with any of it, and just gets like the very basics wrong, and is like seemingly not even willing to put in the time or effort to even try to read and understand these studies himself before he just debunks them for clout on his YouTube channel. So
This paper is garbage. And if the poor YouTuber used this as an example, the poor guy, I mean, it's just sad. It's just sad what they've done to that young man's mind.
But yeah, according to James, what's really sad is what these mean origin of life researchers have done to this young man's mind.
If you do not believe in the physical resurrection of Jesus Christ and you want to hear about why I believe, send me an email to tour@drjamestour.org and we'll get together by Zoom and I'll share with you why I embrace the resurrection of Jesus Christ.